So my name is Iiris Hovatta, and I work at the Department of Biosciences at the University of Helsinki. My lab is interested in the genetic and neurobiological basis of anxiety disorders. And we do a little bit of human genetics work, mostly through collaboration with other geneticists working on panic disorder, for example. But we also work on mouse models of anxiety-like behavior. And my lab is especially interested in the effect of genetic background on stress-induced anxiety and how that influences our identification of potential targets for therapies for human anxiety disorders. So I'll be presenting our work on a mouse model where we have looked at stress-induced anxiety-like behavior in several different inbred mouse strains. And why this is interesting is that we have noticed that the different mouse strains, they respond to stress in very different ways. They have different behavioral response to stress, but also their brain transcriptomic response to stress is very different depending on the genetic background. And now if we think about this in terms of human anxiety disorders, all of us humans, we have a very different genetic background. And it's important for personalized medicine to understand how the genetic background influences what happens in the brain when we get stressed, and also how we should then treat stress-related disorders in an individualized basis. So in this year's meeting, there has been many sessions on stress and stress-related psychopathology. And I think it has become an interesting field of study because we have now started to understand a little bit the molecular mechanisms that happen in the brain when people are exposed to psychosocial stress, and how those particular molecular responses might lead into development of, say, anxiety disorders or major depressive disorder. So I think now the field is really coming together from both the psychological and molecular perspective. And in that sense, this has been a great meeting because there has been a lot of work on that aspect. Also combining studies of epigenetic differences within different individuals and also in different mouse models, and identifying those kind of mechanisms, how environmental factors affect psychopathology. My introduction to the field actually came in 1992 when I was an undergraduate student of genetics at the University of Helsinki. And I wanted to have a summer job. And I went to talk to late Leena Peltonen, who was a leading geneticist in Finland. And she happened to have a job available in the project where she was studying molecular genetics of schizophrenia. This was a big project that was just starting out, and I happened to be there at the right time. And she recruited me to that project, and I finally ended up defending my PhD in 1998 of that topic. So it was partly serendipity that led me into psychiatric genetics. So about the specific sessions, I really enjoyed the plenary session given yesterday by Professor Dan Geschwind, who is studying the transcriptional response to of the autism spectrum disorders, and his group has been pioneering transcriptomic approaches to study the pathogenetic mechanisms behind autism spectrum disorders. And I think his presentation was a great example of how, by combining different levels of genetic information in a systems biology approach, you will be able to find very tractable changes that happen in autism spectrum disorders and that are probably going to lead into better understanding of the molecular basis of that disorder. So this is my first time at the ECNP Congress, and I'm very excited to be here, especially because I'm a geneticist by training, and this is a meeting where people from different fields meet each other, and And I've been particularly excited about the ability to meet a lot of psychiatrists who actually treat anxiety disorder patients and do more clinical research. And that has been a very valuable experience for a basic scientist like myself.